Welcome to Study Loop.
Hi everyone, my name is Eric and let's answer problem 25. In problem 25, we are focusing on the 5' methyl guanosine. Okay, and this is added to an mRNA. So first off, let's go ahead and define what this 5' methyl guanosine is. So this has to relate with the pre-packaging of the mRNA. And so we are going to be modifying this mRNA in order for it to be shipped out from the nucleus out into the cytoplasm. So this has to do with making the mRNA, and typically that is going to be located in the nucleus. So if we pretend that this is the nucleus here, this nucleus is where our DNA is located, right? So our DNA. And we are going to be making our mRNA, right? So this will be our mRNA strand. Out here is going to be the cytoplasm. And the cytoplasm is where the ribosomes are located. These ribosomes are able to take that mRNA to make protein. And then that protein will have function to do something in the cell. And in order for this mRNA over here to be exiting this nucleus, it has to first cross the boundary of this membrane nucleus. And in order to do so, it needs a few modifications so that it doesn't get degraded. One of the modifications that is made is the addition of a 5' cap and additionally a poly A tail. Okay. Now this one is a 5' and this is a 3' poly A tail. Okay. And the way this works is that this 5' methyl guanosine is the 5' cap that is found at the front of the mRNA. This allows the mRNA to exit and cross this boundary, this membrane here. Okay? And that prevents the degradation of this mRNA because the machinery, the proteins, will recognize this 5' cap and recognize that it is an mRNA, and therefore it won't degrade this molecule so that it can be translated with the ribosomes. And so the question is asking, what happens if we don't have this 5' cap? Well, if there is no 5' cap to protect the mRNA, then the machinery will not recognize that it is an mRNA, and they will degrade the mRNA. So the mRNA will not make it out into the cytoplasm, and it won't be able to cross the membrane because there are going to be nucleases that are going to degrade this mRNA. The answer choice that most resembles this situation is answer choice A, where the transcript is going to be degraded when the mRNA is going to try and move out of the nucleus into the cytoplasm. Okay, so this 5' cap is important in the recognition sequences so that it will be recognized as an mRNA and not degraded. To further help you understand this question, let's go over the other choices. So in choice B, we have the mRNA molecule, and it says that it would be stabilized. The mRNA, the 5' cap does play a role in the stabilization, but the translation process does not take place inside of the nucleus. It takes place outside in the cytoplasm. So B is not the correct answer. In choice C, we see that the mRNA would be moving out of the nucleus, but it would not create more copies of the mRNA molecule just because the 5' cap is not present. 5' cap is only for recognition. So C is not a correct answer. And finally, let's look at choice D. The mRNA molecule would not be able to add a poly A tail to the 5' end. We know that the poly A tail is added to the 3' end, and the cap, or the methyl guanosine, is added to the 5' end. So letter choice D doesn't make sense. Therefore, all we're left with is letter choice A, where it would be degraded if it did not have the 5' methyl guanosine. I hope this helps explain the process a bit and the modifications that are made to the mRNA. Thanks for watching, and I'll see you next time.